Hey everyone, welcome to The Explainer. Today, we're diving into something that literally shifted my entire perspective on Parkinson's disease. You know, when we usually think about this condition, our minds immediately jump to dopamine, right? Tremors, movement issues, the classics. But today, we're taking a completely different angle. We're going to look at the brain as this highly delicate, intricately balanced ecosystem, one that unfortunately falls victim to a devastating misunderstanding. We're going to explore the powerful and honestly, sometimes heartbreaking role of neuroinflammation.
There's this quote that I think really perfectly sets the stage for our journey today. The immune system is a double-edged sword. Its fierce devotion can sometimes become the very agent of its own destruction. And that's exactly what we're unpacking. It's a story of fierce devotion gone horribly wrong, where the very mechanisms designed to protect our brain inadvertently become the main drivers of its breakdown.
To really understand this tragedy, we first need to meet our hero, so to speak, the microglia. These are the primary resident immune cells of the central nervous system. Think of them as these highly sensitive sentinels and, well, caretakers. In a healthy, happy brain, they're constantly surveying their environment, gently clearing away debris, supporting the neurons, and just keeping everything in perfect, peaceful harmony.
In the specific environment they're tasked with protecting in our story is the substantia nigra. Picture this region of the midbrain as an incredibly beautiful, delicate neural garden. It's absolutely rich with dopaminergic neurons. Those are the exact cells that pump out dopamine and let us move fluidly and effortlessly. But here's the catch. These neurons are remarkably sensitive to stress. They rely completely on the microglia to keep their garden pristine and safe from harm. Now, what's really interesting about this dynamic is that microglia are essentially shapeshifters. Depending on the signals they pick up from their environment, they adopt totally different personas. On one hand, you have the peaceful M2 phenotype. This one expresses a protein called CD206, and it secretes anti-inflammatory factors to promote healing. But on the flip side, if they sense a severe threat, they instantly transform into this aggressive M1 phenotype, marked by CD16/32. In this state, they drop the gardening tools, pick up weapons, and become soldiers, blasting out pro-inflammatory signals to launch a full-scale attack.
Part 1. The trigger, a polluted garden.
So, you might be wondering, what exactly causes these peaceful caretakers to snap and become aggressive? Well, the disruption usually starts deep down inside the cells themselves. Sometimes it kicks off with genetic flaws, in genes you might know like PINK1 or GJ1. Normally, these genes help clear out damaged mitochondria in a process called mitophagy. But when that clearance fails, we get mitochondrial dysfunction. Suddenly, these failing energy factories start polluting the cellular environment, leaking massive amounts of reactive oxygen species, or ROS. This oxidative stress is essentially a catalyst. It causes a naturally occurring protein, apha-synuclein, to misfold and clump together into these toxic tangles. Basically, our beautiful garden is now heavily polluted.
And this pollution triggers a truly tragic chain reaction. First, our caretaker microglia spot these toxic alpha-synuclein tangles. Naturally, they rush over and attempt to clear the debris. Actually, scratch that, they try to literally eat it, which is a process called phagocytosis. But here is the fatal flaw. These protein clumps are incredibly stubborn, and the clearance just flat out fails. Frustrated and honestly overwhelmed by this persistent threat, the microglia abandon their healing M2 state. They transform into that aggressive M1 phenotype we talked about. Since they can't clean the mess up quietly, they sound a massive chemical alarm, releasing these inflammatory signaling proteins called cytokines.
Seeing how this all connects brilliantly illustrates the core problem. If you trace the path, it goes right from defective mitochondria creating a storm of reactive oxygen species, which then directly fuels that misfolding of alpha-synuclein. Eventually, those form the dense Lewy bodies inside the degenerating neurons. And the worst part? The accumulating synuclein and the ROS continuously provoke the microglia. It traps the brain in this vicious, inescapable, self-sustaining loop of chronic inflammation. Part 2. The maladaptive response. Friendly fire.
All right, this brings us to the real climax of our cellular tragedy. Once they're locked into that aggressive M1 state, the microglia pull out the heavy weaponry. We're talking pouring out pro-inflammatory cytokines like IL-1 beta, IL-6, and TNF-alpha. They're secreting nitric oxide, which just drives the oxidative stress even higher. And all of this creates a state of what we call sterile inflammation. It's an immune attack where there isn't actually a bacteria or a virus to fight off. The tragic result? Those delicate dopaminergic neurons in the substantia nigra, which are uniquely vulnerable to this exact kind of stress, get caught right in the crossfire. They are inadvertently killed by the very cells that swore to protect them.
It is a heartbreaking biological irony. The protectors, in their desperate, confused attempt to save the neural garden, literally become the destroyers. Let the weight of that sink in for a second. Massive neuronal loss, driven purely by a misguided immune response.
Part 3. The climax. Breaching the borders. Let's see how this builds, because the story absolutely doesn't end in the substantia nigra. As these neurons die off, they spill their internal contents, stuff like mitochondrial DNA, right into the extracellular space. And to the immune system, these look like danger-associated molecular patterns, or DAMPs, basically blaring red sirens. The microglia sense these DAMPs and scream even louder for backup. This intense signaling actually does something incredible and terrifying. It compromises the blood-brain barrier. It breaches the very fortress walls that are supposed to keep the brain completely isolated from the rest of the body's immune system. And with the borders breached, it's total chaos. Pro-inflammatory cytokines from the peripheral bloodstream start crossing directly into the brain. Peripheral immune cells, like monocytes, rush in and immediately adopt an inflammatory phenotype. T lymphocytes, specifically CD4+ and CD8+ T cells, just flood the site. Some of them even start recognizing and attacking the alpha-synuclein directly. What started as a localized, contained little fire in the midbrain has now erupted into a full-blown systemic inferno, putting neurodegeneration into hyperdrive.
Part 4. Whispers from the duct. Okay, let's dive into this, because prepare yourself for a massive twist. Scientists are now finding that the origins of this whole inflammatory storm, they might not even start in the brain at all. What completely blew my mind is that in Parkinson's, the gut microbiome often shifts toward pro-inflammatory bacterial species. This spikes the production of endotoxins and drops our protective short-chain fatty acids. The gut gets inflamed, intestinal permeability increases, hello leaky gut, and inflammatory mediators seep right into the bloodstream. But get this, it's increasingly believed that alpha-synuclein misfolding might actually kick off in the enteric nervous system of the gut, triggered by this local inflammation, and then physically travel up the vagus nerve, like a highway, straight up into the brainstem.
A whisper in the gut triggers a storm in the mind. Think about that for a second. It's a profound paradigm shift. A distant, seemingly quiet imbalance in the gut can ultimately trigger a devastating storm of neurodegeneration. It just goes to show you, the body is truly one deeply interconnected system.
Part 5. A new therapeutic dawn.
Okay, so we've covered a lot of cellular tragedy today, right? But here's the upside. Understanding exactly how this ecosystem breaks down gives us incredible power. The ultimate question we have to ask ourselves now is this. If inflammation is driving the destruction, can we step in and calm the storm before the delicate neural ecosystem is irreparably broken?
So, the crucial point here is that science is pivoting hard. Instead of just trying to replace the dopamine after it's lost, researchers are actively advancing immunotherapies right this second. Imagine detecting the disease years before motor symptoms even appear, maybe by spotting microglial activation in patients who just have early warning signs, like REM sleep behavior disorder. We're rapidly developing specific antibodies to clear out that toxic alpha-synuclein before the microglia get frustrated. We're looking at drugs to actively suppress that aggressive M1 microglia response. We're even exploring how to use regulatory T cells to step in, act as the peacekeepers, and quiet the immune attack to protect the surviving neurons.
This is a monumental shift in how we view Parkinson's disease. We're moving way beyond simple symptom management now. By targeting the immune system, we are finally learning how to restore the peace in the neural garden, rather than just endlessly trying to replace what was lost in the war. It's the pursuit of true, actual disease modification.
I want to leave you with this final thrilling thought. If we can learn to listen to the body, to catch that inflammatory whisper in the gut or the blood super early on, and if we can reprogram those powerful sentinels of the brain back to their healing state, could we actually stop neurodegeneration before it truly begins? The science is moving incredibly fast, and the hope on the horizon is very real. Thank you so much for joining me on this deep dive. Keep asking questions, and I'll catch you in the next explainer.